crashing the party, but I think it's pretty cool that we can all work together. It is an absolute pleasure to present to you a project, I guess, that we're doing that follows the lines, I guess, initially set by GenTORE. I don't have to go through the definition of resiliency, I think, in this crowd. I looked it up at the very beginning of the project, and I try to keep it in front of me at all times to remember that we're not compressing or deforming our cows. We're just looking at which ones are able to recover or easily adjust to adversity or change. So we're not squishing cows here. We just need to keep that Merriam-Webster definition in our minds. This is a graph showing the change in the number of cows in Canada since 1960. The blue line shows the change in the number of cows, and the dark blue shows how much milk is produced. And what you can see here is that 1/3 of the cows are producing exactly the same amount of milk. And I think that that's pretty resilient. That's a pretty resilient industry, especially given the bad rap that we all know cows currently might have. We talked a lot about that in the plenary session, but I think that overall the industry is quite a resilient one up to now. But it would warrant a closer look at the individual cows. So we thought about a definition for what a resilient cow could be. Given the previous definition, I think we have to include in the definition of a resilient cow an animal that's able to adapt rapidly to changing conditions. But in the context of food security, we also have to think about an animal that doesn't compromise its productivity, health, or fertility. And in the context of the environmental changes that are happening right now, we also have to have animals that are becoming more resource efficient and overall reducing their environmental burden. So this is our definition of resiliency from a Canadian standpoint. This is what we're aiming for. So as a geneticist, what am I supposed to do about this? Well, we know that genomic tools have been used to select for fertile and healthy feed-efficient cows, and it can be used to decrease methane emissions, but we need really high-quality phenotypes to achieve this goal, and that's a real challenge. So our solution is similar to GenTORE, to work with local populations and link with international efforts. As well, another really important aspect of this is also to collaborate with other researchers in related fields. We have to stop siloing ourselves, and we have to start sort of broadening our horizons to work with maybe even a veterinarian now and then, even though we don't really get along usually. So just a little bit of history from our Canadian perspective. In 2015, Filippo Migliore and Flavio Schwenkel and Paul Stothard started the Efficient Dairy Genome Project. Actually, Flavio came into that afterwards, but he brought it over the finish line. So what the goal of this project was to develop an international database for feed efficiency and methane emissions. And it was a very successful project. Many of you have been involved in that project. We finally launched our feed efficiency evaluations officially in Canada in April of this year. So that was the goal of the Efficient Dairy Genome Project. But in the course of that project, we realized that efficiency is not everything. And we really need to add some of these aspects of resiliency as well. So we have started a new project and thankfully got funding for it, where we're aiming to collect closer to biology fertility traits. We're looking at new health trait evaluations, further analyzing feed efficiency and methane emissions, and even, crazily enough, we're going to try to put in some epigenomic data in a routine evaluation. Now, whether or not that works out is still in the stars, but it's on the list of things to do. We're very fortunate to have the same partners as the Efficient Dairy Genome Project: Australia, Canada, Denmark, Switzerland, and the USA. But we're also very happy to have added Brazil, Spain, and Germany to that list. So within Canada, we've also tried to incorporate other groups into our research project. We span with universities from the East Coast in Prince Edward Island all the way through to the West Coast in British Columbia. But I think even more impressive are these international collaborators with whom we have the pleasure of working with. And to that, I do applaud Nick for participating in a lot of our discussions, even though some partners may or may not be able to provide a lot of data. But you can see that we have a really great group of countries and groups working together to attack this problem. So our project, just briefly, has 8 activities. We have a fuel tank, sort of, that's consisting of phenotypes, where we're looking at closer to biology fertility, enhanced disease resistance, and feed efficiency and methane reduction. This fuel feeds into an engine where we're doing activities that are revolving around genomic and environmental relationships, multi-generational effects and epigenetics, as well as, of course, a data management plan. As with every good Genome Canada project, for those of you who are familiar with Genome Canada, we are required to have a genomics and its ethical, environmental, and economic, and legal, and societal aspects. So that's what that GELS there stands for. But it's really a neat thing to have in a project like this because it forces the natural scientists to think about socioeconomics and how we can maximize sustainability and societal acceptance. Finally, of course, the goal is to translate and implement the results of this project and ultimately result in genomic tools to select for sustainable cows. So how will we do this? It's a big list of things to do. The first part, of course, is being run by Dr. Ronaldo Seri at the University of British Columbia, and he's a reproductive physiologist that is aiming to standardize phenotypes based on automatic sensors. Now, in the genetics world, I know a few geneticists have tried to do this, but I think it's really good to have a real reproductive physiologist at the helm of this boat. He's already got some really nice results that he's published, and we're really trying to get close to the physiological factors affecting estrus expression and embryo survival. Ultimately, the goal here is to identify genomic markers of estrus expression and fertility and incorporate them into a national evaluation. The second activity is being run in Guelph, and we've already had our first check off the list of things to do. That's fertility disorders. That's been included in routine genomic analysis by Lactanet in December of last year. We're also developing methods for routine phenotyping of Johne's disease or paratuberculosis, calf health, and leukosis. And as many of you can already imagine, animals that are, for example, affected by leukosis, Johne's disease, their later somatic cell score is also negatively affected. So on the graph here, you can see animals that have been identified as either being positive or suspicious. They have a higher cell score than animals that are that have a negative average for Johne's disease. Feed efficiency and methane emissions, as I said, that's really where the heart of the collaborations is happening, and we're enlarging our reference population to an expected total of approximately 17,000 animals collected for feed efficiency and just under 8,000 animals for methane emissions. Some of the challenges with regards to this are, you can see some of the countries, just a schematic diagram of what the data looks like from the different countries. And you can see that there's quite a few differences even in how people are recording their trait, whether that be dry matter intake or methane emissions. And this is just one of the many challenges that we're going to have to deal with as we move forward. But I feel confident that we'll figure out how to do that. Makes for a nice picture anyway. Dr. Flavio Schenkel is running activity 4, where he's looking at genomic and environmental relationships. In particular, he's using multi-trait GWAS and different meta-analyses to identify genomic regions, possibly with pleiotropic effects on all of these resilience traits. You can see that on the graph in blue, that the effects or even the parameters for a lot of our efficiency traits are indeed dynamic over the course of the lactation, so that has to be taken into account as well. Flavio is also working on identifying copy number variants that have an effect on resilience and investigating the effects of heat stress on important traits. That work is being done by Ivan Campos. Excuse me. Marc-André Sirard from Université Laval is running the multi-generational effects and epigenetics portion or activity of this project. This is the secret one. This one you're going to have to wait another year or two to have any results at all on, because it's really difficult to do. But ultimately, his goal is to quantify the effect of early environments on the resilience of daughters. And he's surveying for epigenetic signatures on very, very precisely phenotyped animals. Dr. Paul Stothard of the University of Alberta is running the project database, where he's looking at whole genome sequence data and creating a genome browser and integrating the GWAS findings and the epigenetic signatures and annotated sequence variation from the other activities in the project. Dr. Ellen Goddard and Dr. Getu Hailu are doing the socio-economic analyses that I spoke about earlier. These two are working with Statistics Canada to analyze how farmers actually make decisions about trade-offs between traits. They're also looking at the farm-level outcomes from selection of resilience traits and what's going to happen to the market-level outcomes from selection of resilience traits. And finally, what I think is really interesting is the public acceptance of dairy under different breeding strategies. One of their earliest research results from the previous project was that when you're speaking to farmers and you would mention the word methane emissions, they would shut down. But if you mention feed efficiency, they would get all excited. But when you're speaking to the government, you have to speak about methane emissions and definitely not about feed efficiency. So it's a really interesting play, and now that we've got publications saying this, we can all take trust in what we actually now know. We've thought about it for a long time, but now we know it. Activity 8, the final activity, is being run by Dr. Gerrit Kistemacher of Lactanet. That's the National Evaluation Centre for Genetic Evaluation of Dairy Cattle in Canada. And he has the lofty task of developing the Resiliency Index. We did implement, as I mentioned, the fertility disorders evaluation in 2020. The feed efficiency index came out in April of this year, and our next goal is to develop this resiliency index. As many of you may know, the Canadian feed efficiency trait is split into 2 stages, where we have 3 traits in the first part of the lactation and 3 traits in the second part of the lactation, and that constitutes our feed efficiency evaluation, aiming to select for cows that use less feed at the same level of production and body size, but after the peak of lactation. So in summary, dairy cattle resiliency is really a top priority, but we require really high quality phenotypes. And as Mike Coffey loves to say, in the age of the genotype, the phenotype is king. I hate to say it, but he's right. We need national, international, and interdisciplinary collaborations to run projects and programs like this in order to have any chance of success. And as you can imagine, there are really a lot of moving parts in projects like these. And Nick, I'm sure that you can appreciate that as well. So we have a lot of next steps. You can have a look at them, but ultimately, bringing all of this from a research sort of point all the way into a national evaluation is a very lofty task. I know many of you are researchers, but some of you are also working at national evaluation centers, and there is a lot of collaboration and discussion and communication that has to happen before these ideas actually get turned into tangible breeding programs. And I think all of us are really lucky to be a part of that. And with that, I would like to acknowledge all of the institutions and organizations that are taking part in our Resilient Dairy project. If any of you are interested, please have a look at the website there. And last but not least, I of course have to give a huge shout out to the fantastic team of people who make this all possible. These are just a few of the students who happened to show up at my house one evening, but you can imagine that there are quite a few more involved in this. Thank you so much.